If you watch nature programs on television, you know there seem to be lots of dangerous, ferocious creatures out there. Man-eating crocodiles, woman-eating sharks, deadly snakes. It's enough to make you want to stay inside in air-conditioned comfort. But is that all there is? And how do we know? Well, here's where we find out. Hi everybody, I'm Charles Messing, and we're here to explore the incredible diversity of life on Earth, from bacteria to beetles, from mildew to mandrake, from amoebas to zebras. Now, in preparing for this show, I wanted to get a handle on what you probably know about so I could figure out what you probably don't know about and concentrate on that. So I did what any self-respecting biologist would do. I grabbed the remote, a nice Cote d'Aron, and sat down to do some serious surveying of nature TV. Here's what I found. Mammals accounted for about 35% of what I saw. Reptiles and amphibians, about 15%. All of the bugs, the spiders and insects and centipedes and so on, about another 15%. Birds, about 11%, and fishes, about 7%. That totals 83%. Everything else, only 17%. Okay, so what this says is that you basically know about creatures with teeth, fangs, claws, tusks, horns, and poison. Is there anything else? Well, consider the humble yeast, a one-celled relative of mushrooms. It's not sexy. It's not glamorous. It's not even particularly dangerous, although there are those yeast infections. Hell, the silly things can't even move. But without yeast, we would not have bread, beer, wine, or cake. And you know what that means. If Marie Antoinette had said, "Let them eat crepes," the French Revolution might never have happened, and we'd still be using the old, outdated English system of measurement instead of the far more logical metric system. Come to think of it, we are still using the old, outdated English system of measurement. Anyway, there you have it. The humble yeast, pretty good for a microscopic fungus. We just showed you on our Nature TV survey what percentages of species appear on television. That doesn't necessarily mean those percentages reflect how species are divided up in real life. Just because we see mammals 35% of the time in nature programming doesn't mean that mammals make up 35% of the 1.7 million species of living things on the planet. In fact, the actual numbers in real life are quite different. As you can see here, the mammals, birds, reptiles, and fishes together, that is the vertebrates or animals with backbones, just like us humans, only make up 3% of species on Earth. Now, what groups of creatures are more diverse? Well, let's ask the Haggertys from Beloit, Wisconsin. So the question is, what groups of living things have more species than the animals with the backbone?
Well, Professor, we vacation all the time at the beach, and our son David's been collecting shells since he's been a little one. I'm going to go with seashells.
Seashells, that would be the snails and clams, cockles and mussels, alive, oh, the mollusks. And don't forget the octopus and squid. Survey said, yes, over 80,000 species. So, you're on the board, let's see what you can do.
Well, I've been gardening for years, and I'm going to have to go with plants.
Plants, everything from gardenias to giant redwoods. Survey said, yes, over 250,000 species. So, young man, let's see if we can take home a victory for your family.
How about germs? Bacteria.
Bacteria, lots of them out there. Survey said, oh, I'm sorry. lots of bacteria in the world, but only about 9,000 species have been described. So let's go over to the barbers from Houston and see if they can get on the board. So,
Howdy, Professor.
howdy. So here's the question. What groups of living things have more numbers of species than animals with backbones?
What with all the humidity we got down in South Texas, it's a constant battle against mold and mildew, so I think I'm going to say fungi.
Fungi, let's see. Mold, mildew, and mushrooms, survey said. Over 100,000 species. Good answer. Very good answer. So you're up on the board. What's your answer?
We also have all these creepy spiders and scorpions.
Arachnophobic, huh? Arachnophobic. Arachnids. Let's see if they're up there. Yes! 75,000. Good answer. Good answer. Only one more for a victory here.
Okay, I'm gonna have to say bugs, insects.
Insects. Good answer. Butterflies, moths, beetles, ants, wasps. A long list. Survey said. Yes! Over 950,000. Congratulations. Congratulations. Very good. In fact, that may just be scratching the surface. Go to a tropical rainforest or a cloud forest like this one outside Baños, Ecuador, and there are probably enormous numbers of never-before-seen insect species, probably mostly beetles. In fact, more than a quarter of all known insect species are beetles, over 350,000 described so far. Let's see if I got anything. When the great geneticist J.B.S. Haldane was asked what his study of the living world had taught him about God,
"The Creator, if he exists, must have an inordinate fondness for beetles."
Some scientists believe that we may only have described 10% or even only 1% of the kinds of living things. Consider that we recognize about 20,000 species of nematode roundworms. In addition to occupying almost every square inch of mud and soil on Earth, often by the hundreds like these, nematode roundworms also live as parasites in pretty much everything else, including fish, plants, insects, and people like these. Every species that you can see with the naked eye might have at least one of its own roundworm parasites. So right off the bat, we might nearly double the known number of species. I want to give you an idea of how much diversity we know exists, even where you don't really expect it. So we need to take a short trip to a deceptively empty place, the Gulf Stream. We're here about 5 miles off the coast of Miami, Florida, aboard the research vessel Coral Reef 2. And if the length of time I've been out here without so much as a nibble is any indication, there's not a whole lot out here. So we're going to have to use a variety of tools to figure out what's living out, or rather down here. So let's begin by trying to find out if the water is as empty as it looks. To do this, we'll use a Niskin water sampler, which is basically a hollow tube that we send down to pretty much any depth we want to and trigger it shut by this brass messenger. Voila. Now we can't expect to just look at this water and see anything. What we have to do is treat the microorganisms in here with a chemical that will stain their DNA and make it visible to us under fluorescent light. In order to capture larger things that may swim more rapidly, we need a bigger net, and we need it to have a larger mesh so that we can pull it through the water more rapidly. This is an Isaac's Kidd midwater trawl. So we're going to deploy this in about 300 meters of water. As the dusk settles, small creatures from deep water come up towards the surface at night. We hope to get some really interesting things. One of the great things about exploring the deep ocean is that you're never sure what you're going to come up with. Unfortunately, one of the not so great things about exploring the deep ocean is that it's deep. So it sometimes seems like it takes forever to get the net down, to tow it long enough to get a good sample, and to get it back up to the surface again. Surface! All right, let's see what we have here. Oh, great. That's a tornaria larva. You can see its cilia going. It's going to grow up into an acorn worm. And that pink copepod has got a cluster of green grape-like eggs at its hind end. There's a fish larva over there. And, here, oh, this is a shrimp larva. That's great. It's still kicking. And it's got a little something there in its mouth. Oh, and this is Sapphirina. This is an incredible copepod, flat as a thin piece of cellophane. Let's see, take a look here. Oh, the colors are not part of the copepod. They're refracted and diffracted light. These things put on an incredible display when the sun hits them in shallow enough water. And we haven't even begun to talk about the larger things, the mackerels, tunas, sailfish, marlin, sharks, whales and dolphins, or even the mid-sized fishes like the lanternfishes and hatchetfishes and things like that. So there's a huge diversity out here we haven't even begun to see. So okay, we're claiming that there are millions of species of creatures on this earth and we keep finding new ones. But how do we decide if we've got a new species or not, whether it's a crustacean, a bird, a fungus, or an arrowworm? How do we know? Well, to cut to the chase, you basically have to ask an expert, a taxonomist, someone who has a major, usually professional familiarity with the group in question. Back a few hundred years ago, when there were a whole lot fewer known species, the Swede Carl Linnaeus, who stylishly Germanified himself as Carl von Linné when he was knighted and gave us the basic system of classification that we still use today,
Kingdom, class, order, family, genus, and the species. Yeah, that's it.
was able to recognize and describe new species of pretty much anything: plants, insects, sea urchins, birds, fish, and mammals. Linnaeus also standardized the two-word system of scientific names, each consisting of a genus and a species. In fact, he was the one who came up with the scientific name Homo sapiens for us, the human species.
Oh, a wise guy.
Well, yes. Homo sapiens means man the wise. But unlike during Linnaeus' time, there are so many species known now that every taxonomist has to specialize. Dr. Daphne Foten of the University of Kansas works on sea anemones and sometimes hunts for them in unlikely places. You know, Toto, I don't think she's in Kansas anymore.
Wow, this is the place for them.
Dr. Andrew Rogerson of Marshall University studies microscopic amoebas. He calls them amoebae. Me, I study the crinoids, the sea lilies and the feather stars. They may look like plants, but they're actually animals related to sea stars and sea urchins. They have the same 1, 2, 3, 4, 5-sided construction and so on. Now, many taxonomists know their critters so well, or the unknown specimen is so different from everything else, that sometimes, right off the bat, you can tell that nothing like it has ever been seen before. That was the case with the megamouth shark, first discovered in 1976. Though up to at least 18 feet long, it is harmless and uses its numerous tiny hooked teeth to feed on plankton. But it was so different from the other giant plankton-feeding sharks, the whale shark and basking shark, that the scientists who described it placed it in its own family. It would be as if the only carnivorous mammals you knew about were dogs, wolves, foxes, skunks, bears, raccoons, weasels, and seals, and all of a sudden someone discovered lions, tigers, and cats. That's how different it is. So even when you're confronted with something that's really new and different, a taxonomist has to first dig into the books and articles that describe what is known. Your popular field guides won't help you much here.
Ow!
So you have to go to what we in the taxonomy game call the primary literature, where the species were originally described. For my critters, the crinoids, that means just a little bit of light reading to wade through. Then you compare your specimens with the original descriptions. You'll have to take measurements, counts, make drawings, maybe even use an electron microscope to look at the really small bits, all to see if your guys are really different or not. But sometimes that's not enough. Sometimes the published descriptions just don't have enough details, especially if they're older ones. So you have to go back to the original specimens, and that usually means a museum. Museums, like the Natural History Museum here in London, are libraries of the world's biodiversity. They archive much of what we know about the natural world. This is where they keep the good stuff. The Natural History Museum has an excellent crinoid collection, including specimens dating back to the original HMS Challenger, the first deep-sea, round-the-world oceanographic expedition over 130 years ago. But more importantly for us, it also has a great type collection. Whenever a scientist describes a new species, he or she designates one specimen as the holotype and usually one or more other specimens as paratypes. The holotype becomes the ultimate arbiter of what defines the species, while the paratypes give you some idea of the range of variation. Now, on the one hand, it may seem ridiculous to have a single specimen stand in for the variation of an entire species. It's an idea that's actually completely antiquated and obsolete. It dates back to Plato, who thought that what we saw in the real world was only a poor reflection of the true ideal essence or eidos of a thing. It took almost 2,000 years before Charles Darwin came along and turned that idea on its head. He saw that it was the variation among individuals of the species that was important, not some ideal essence. But we still need the holotype in case the original description turns out to be vague or inaccurate, or one or more of the paratypes turn out to be a different species. So you examine those museum specimens in detail. You take more notes and measurements, maybe make some more drawings, to compare the museum specimens with yours. And with any luck, you discover that yours are really different, that you do apparently have an undescribed new species. Of course, for most folks, one crinoid pretty much looks like another. So let's take an example that's a bit bigger and more obvious. Everyone knows that there are two species of living elephants. The African elephant with the big ears, flat forehead, and concave back, and the Asiatic or Indian elephant with the small ears, the bulging forehead, and the convex back. I bet you never noticed that business about the foreheads and the backs before. Anyway, where on earth do you hide a third species of elephant? Well, it turns out right under our noses. When scientists developed an elephant DNA identification system for tracking the sources of poached ivory, they discovered that there were actually two different species of elephants in Africa: the well-known savanna elephant and the less well-known forest elephant. Scientists used to think that the smaller, stockier forest elephant with its round ears, its darker color, and its straight pinkish tusks was just a geographic variety or a subspecies of its larger cousin. But the great differences in their DNA suggest that the two might be as different as lions and leopards. But whatever you're working on, elephants, crinoids, or mushrooms, you sit down, gather your notes, your photographs, your drawings, your DNA data, your reference sources, and you write a description. But what to name it? Well, you could name it after someone, say the person who collected it. Linnaeus named the black-eyed Susan Rudbeckia hirta after his patron, Olaf Rudbeck. Or you could name it after the place it was found, like this helmet conch, Cassis madagascarensis, named for the island of Madagascar in the Indian Ocean. Of course, that becomes a little less useful when you discover that the thing is found from North Carolina to the Caribbean. The best thing is to give it a descriptive name, one that reflects some feature unique to your particular species. Like this, Xiphias gladius, the broad-billed swordfish. Tamen, vos es inconcessus utor just. Sorry, but you just can't use any word you like. First of all, it has to be in Latin, or if it comes from a Greek word like Tyrannosaurus, or a Chinese word like Huayangosaurus, you have to treat it like a Latin word. Kind of like the way we treat French rendezvous, Russian Sputnik, or Japanese sushi in sentences like English words. Now, why Latin, you may ask?
Is that because of the toga parties?
No, because Latin is a dead language. Nobody gossips or blogs or buys groceries in Latin. The point is that Latin word meanings do not change. There's never any new slang, and no words become obsolete. And Latin also levels the playing field. Every scientist from Mongolia to Malaysia to Montana has to learn the same name for the same species. Now this is a gladius. Now, once you've got your manuscript completed, you've got to get it published. And there are numerous professional scientific journals just for that purpose. Some, like the Proceeding of the Biological Society of Washington, are pretty general. This issue has 2 new species of bats, a description of a new fish, a new snake, 3 new crustaceans, and a new worm. Others, well, like Crustaceana, they speak for themselves. The rest is pretty much standard operating procedure for modern science. You send your magnum opus off, the journal editor sends it out to experts for their peer review, you get it back, eventually, with changes, comments, and recommendations and suggestions from the reviewers. Of course, one of the reviewers might gently have suggested that what you thought was a new species actually was already described in some obscure German journal 157 years ago that you knew nothing about, in which case you've wasted your time and everybody else's, but that's not as much of a problem as it used to be. In any case, there's no international board of judges who decide in advance if your species is valid or not. When it's finally published, it becomes another brick in the wall of our understanding of life on Earth, ready to stand or fall on its own merits. Who writes this stuff? So there you have it, the diversity of life in a nutshell. I hope I've given you some idea about what's really out there and how we find out about it. In upcoming shows, we'll discover much more about life's variety, how creatures have adapted, how they interact, who's related to whom, and how they've changed over time. Now, if you'll excuse me, I've got to go see a man about a synonym. A synonym is about what you'd expect. 2 different names that have been given to the same species. The rules say that the older one gets priority, but some folks need a little reminding.